Hello, everyone. We'll be starting here in just a few more minutes, so thank you for joining, and please be patient. We have another 5 minutes until we start.
Hello, everyone. Thank you for joining. We will be starting in a while.
All right, let's get started. So, Anika, I think we can begin.
Yes, Ilya, I think we can begin and others can join in the meantime.
Okay, welcome everyone to this webinar, and thank you for joining today. Today's topic is data science, OmicsLogic data science. So how is data science different from bioinformatics? And what we typically speak about is bioinformatics and application to different types of data or to different domains, biomedical data. Today, we are going to be talking about the more technical aspects of this upcoming program and how you can benefit from actually gaining a lot of the technical skills in coding, in statistical analysis, and machine learning, and really see the application of these methods, not just at the conceptual level, but actually from learning about why and how to pick specific methods all the way to implementation. So I will be introducing the program, but together with me, Dr. Harpreet Kaur, Clinton Kunha, and Dr. Mohit Mazumdar will be taking on different sessions. And so before we begin, I will just briefly introduce OmicsLogic and what is it that we typically do. So as I mentioned, our focus is on biology as a data science. So we speak a lot about the applications of data analysis to high throughput data and training that we provide to elevate the level of biomedical research and also enable research by providing specific benefits and application to oncology studies, infectious diseases, neuroscience, as well as agriculture. And so one of the connecting dots between all of these are the applications of data science methods. And so today we'll talk a little bit more in depth on that topic. But typically, we provide research enablement through analysis of big data. So we actually have big data analysis tools that are designed to address challenges in processing, analysis, visualization, and integration of different types of omics data and phenotypic data. We provide mentorship where you can interact with our mentor community and actually get one-on-one private mentorship if you need in application to different types of data. And we also provide asynchronous training resources. So we have a collection of courses and programs organized with collaborators and different types of university, high school level, as well as college level training that we provide. So let me go on and briefly introduce this topic of data science. So what exactly is data science? Data science is the ability to work with data, so manipulate data, organize data, and that is informatics. And then to apply meaningful statistical analysis techniques to this data, which include different types of machine learning and statistical analysis and even visualization. So it's really analysis of that data. And then finally, you have this application of these methods and of this data to a particular domain or a specialization. And when we speak about data science, in general, data science tends to be agnostic. So data science methods, just knowing how to run analysis, could be applied to very many different domains: clinical, business, Internet of Things, finance and marketing, biological research. But specifically, if we talk about biological data, there's going to be a specific additional level of specialization that we need to discuss. And that will be the way that data is generated, and therefore, what are the specific questions that are being asked of that particular data set, and how can we later on interpret it? Because as opposed to other types of data, a lot of times biological data is much more challenging to interpret and to understand the technical versus the biological patterns that we find in this data. And those are critical to distinguish as we seek to find interpretable, meaningful insights from this kind of data. So the OmicsLogic data science program is designed to provide an overview of statistical analysis and machine learning methods, to discuss experimental design and some of the technical variation that we need to address before the data is analyzed, how to mine data for meaningful patterns, and what do we mean by associations, and then also the application of complex biomedical or biological systems using big data. So how do we infer these relationships from data, and how do we interpret them in the context of this complexity of biological systems? And so specifically, this program is going to be focused on machine learning for biomedical discovery. Specifically, we'll talk about what data science and bioinformatics are designed to do, and how can you learn the conceptual approaches, but also the practical skills. We will talk about data processing, cleaning, and exploration, the application of data science in biomedicine. So what types of projects and what types of examples are going to be meaningful for this kind of analysis. And then we'll introduce machine learning algorithms for next generation sequencing. We'll talk about data mining in the context of X-omics integration, or the integration between different types of omics data, and how to apply deep learning and clinical phenotype annotation for some of the advanced methods of analysis that are being discussed and sometimes used in the context of omics data. Okay, so I see that a couple of messages here that people are mentioning that they cannot hear. Unfortunately, I think that is probably because of your connection or settings. So if you could try and rejoin, and so Narika, maybe you could post this message also in chat if people cannot hear. So specifically, I think that in the context of biomedical discovery, right, we're talking about the different aspects of biomedical discovery where machine learning and the analytics have to be contextualized, because we are going to be trying to connect the research question or the clinical or biomedical need and challenge to different levels of statistical analysis and exploratory analysis that allow us to connect this to the type of data that is being generated that essentially represents some kind of a biological experiment, even though this experiment could also be a design of a data collection without a specific experimental condition, but more in the real world sense. And so specifically in the real world sense, what types of data are we typically talking about? So as we move into this era of precision medicine, we typically mean that there is some kind of a phenotype at the top of the iceberg, at the level where we can see and observe the symptoms. And then underneath, we have this molecular data that we need to connect to the clinical data in order for us to be able to integrate the signal and extract some meaningful results. And so obviously in the context of biomedical discovery, we're talking about biomedical applications that include treatment and selection of treatment options, stratifying risks for patients, but also we're talking about the application for drug discovery and for clinical trials. And so in drug discovery, this analysis of big data from clinical data as well as from high throughput experiments can speed up the development and the translation of discoveries to the use by patients. But also in clinical trials, it's an important component to be able to select the right kinds of patients, to define the right kind of strategy for outcomes, and also to translate other findings that are beyond the objectives of the specific clinical trials. In other words, even if clinical trials have failed, to a meaningful insight for the next round of clinical trials or for modification. And so a big trend in both of these is the discovery of biomarkers. And these, of course, are markers of specific conditions, markers of specific biological processes. And we are looking for more and more precise markers. Oftentimes, what that means is that we are not relying on a very clear-cut signal, but a complex signal. And that complex signal is defined by the complexity of omics data, which could be, of course, of many different types and has many different ranges of values that we can use to combine into a signal. And that signal would be a integrated signal essentially from multiple types of omics data. So what kinds of omics data are we going to talk about? We're going to talk about phenomics, which is the characterization and the features coming from image analysis, from clinical data, from cell morphology. We're going to be talking about genomic data, or the mutations or collections of mutations found in the DNA. We will be talking about epigenomic signal, where we'll discuss ChIP-seq data, whole genome bisulfite sequencing data, transcriptomic data, including gene expression, levels of gene expression, transcript expression, and alternative splicing. And we will talk about specific examples where we will use this data to actually discuss the objective and the type of data that could be analyzed, because every type of data comes with a specific set of parameters that we need to consider when we are trying to set up and plan our analysis. And of course, one of the major challenges in this whole field of omics is that the data is actually unstructured, right? So as you look at the availability of data and the type of data that is being generated, more and more, this is going to be unstructured data, because structured data requires processing. So if you think about collection of biological samples from patients, or even generating that data as a high throughput experiment, you end up with unstructured raw reads. You look at different types of environmental conditions. And because of that, the first step that we're going to be talking about is processing that data, or converting that data from raw to a structured format, from different types of biological material that can generate digital data to an actual organized table that we can work with. And so later on, I'll talk about some of the formats that that data is stored in, so that we can discuss some of the more technical structures for this data that we will be looking at. But ultimately, the objective of data science is to provide guidelines for different steps of data analysis, which includes data processing, as you can see right here, exploratory analysis, and the actual data analysis, or comparative data analysis, or sometimes modeling of that data. And so to go from experiments to results, those three steps is what we will have to cover in this program, and talk about data cleaning and processing, formatting it for the specific type of analysis that we would like to do, exploring data using visual methods and tools, as well as looking at different types of approaches for data mining, data analysis, where we consider different statistical, machine learning, and deep learning models, and then inference. How do we actually test our findings and go back to where we started with, so that we can contextualize the findings and actually interpret meaningful results. So what are some of the typical challenges in this process? Some of the typical challenges include analysis and visual exploration of data, where we can use things like principal component analysis or clustering to really identify some meaningful groups of objects based on all of the available data, or based on an extraction of data. Oftentimes, this also means looking at taxonomic relationships, where we can combine biological understanding of where the data came from with our ability to structure different types of groups of data in the patterns that we find. So a lot of the processing specific to biological data actually combines those two methods. One is just exploratory analysis on aggregate of all the data that we have, including noise and signal, filtering for specific signal that is statistically significant, or integration between taxonomy, which is a biological concept or a construct, and the actual analysis of similarity based on the data availability that we have. So because of that, we are really looking at this collection of different types of data that also includes other types of data, and that could include imaging, clinical data, chemical data, phenotypic data, and all of this data has to go through this process of preparation and organization of this data into structured formats, which typically in the context of data science is going to become some form of a data frame. And so the first thing that we will do in this program is we will discuss how to prepare different types of structured data objects, data frames, vectors, matrices, arrays, and lists from the data sources that we prepare in the processing pipelines. And then we will talk about how to meaningfully organize this information so that we can look at relationships between categorical data, continuous data, and other types of data that we need to organize in a visual format so that we can meaningfully spot some results that are of interest for our analysis. So sometimes this means just summarizing the data that we have. Sometimes it's looking at patterns overall. A lot of times it leads us to the need for data preparation. And data preparation means that oftentimes data comes in a format of different statistical properties that we have to change before we actually can run an analysis designed to deal with a certain data distribution. For example, oftentimes when we look at next generation sequencing data for transcriptomics, we need to first transform it into a log normal scale to have a more normal distribution. Of course, this is not a universally applicable method because when we come to data sets that are, for example, metagenomic data, there typically is not a need to make that transformation, but instead we need to choose a completely different method of analysis to extract meaningful patterns. Now, also when it comes to exploratory analysis, we also need to understand that the majority of the data that we have has many different components and many different sources of signal that we could consider, including technical, including statistically significant, and including some non-statistically significant, but nevertheless important for our ability to extract meaningful portions of data for downstream analysis. And so that's why we will also talk about different methods of statistical analysis, including t-tests, f-tests, ANOVA, regression, and other types of methods that will explain the relationship between variables in our data that we can ultimately use for analysis. And oftentimes we will find that the statistical analysis has limitations, and the limitations come from several different types of sources. So one is the variability in our data is complex, and we cannot capture that complexity using standard statistical techniques because of low p-values and kind of complexity of the differences between noise and signal. It also can come from just the types of data, because we might deal with many different types with many different variables that we cannot connect into a single data set. And so that is really where we will turn to the topic of data mining and classification, looking at many different types of methods that are typically used for analysis, for classification, and for feature selection. So we will look at different examples of methods like decision trees, support vector machines, hierarchical or k-means clustering, including clustering for big data sets like PAM, fuzzy, and dbScan. We'll look at linear discriminatory analysis and other types of methods that will help us use this predictive analysis for inference, for feature selection, and something that we can ultimately interpret. We will also dig deep into the algorithmic properties of some of these methods to really understand how different approaches can help extract features that are responsible for discrimination between different types and different classes. And so we'll talk about methods of feature selection, including support vector machines and random forest for feature significance, as well as greedy methods of analysis like stepwise analysis of linear discriminatory analysis and such. We will also talk about an obvious topic that many are interested in, which is deep learning applications in omics data. So we will talk a little bit about different approaches for deep learning, how we can discriminate between different types of network topologies, how we can optimize some of the parameters, and ultimately, what are the applications? Where do these methods work well, and where do these methods do not work well? And how do we evaluate the performance, and how can we select the right type of method for the right type of project idea that we have? Now, importantly, many of these methods come with ready-made packages and with practical exercises that you will be able to run. And so we have prepared a collection of scripts and curated data sets where you will be able to try and run the specific analysis of your choice, understanding the methods and their application in the context of what's called a package that might combine many different specific scripts and algorithms to extract some meaningful insights that is considered to be a standard in a particular field. As you can see here, this is the application of non-metric dimensionality scaling in MDS for taxonomic analysis of microbiome data. So we'll discuss these methods that really tend to combine multiple methods of visualization, of processing, of data analysis, and then translating that into biologically interpretable results. Now, what we will do in terms of your technical skills is we will introduce a number of different techniques and technologies to allow you to understand and appreciate the implementation of these methods. And so for coding, we will use both R and Python. We will introduce you to a number of different environments where you can run this code, including integrated development environments like RStudio or Visual Studio for R and for Python, respectively. We will talk about sharing your code. How do you effectively share your code using Jupyter notebooks, how to post your data on GitHub, how to create Google Colab notebooks that you can share and run your code in those as well. But ultimately, you will get a sense or a landscape of these different methods and tools, including some commonly used libraries and packages like the base in R or ggplot, specific packages like ggfortify and ggpubr. You will be able to look to use Python pandas, NumPy, and sklearn packages, including visualization in matplotlib and Seaborn. And you will be able to apply general coding practices that are going to be important as you develop your skills in bioinformatics. And that includes organizing your scripts using comments and functions, setting up the development environment, dealing with errors and troubleshooting your code. So learning how to interpret the errors and finding the reasons for the errors, and then how to prepare your data summaries and submitting curated data and metadata tables to sharing repositories like on GitHub or Dataverse. And we'll use Jupyter notebooks on GitHub and Google Colab, including we'll create some interactive visualization using Plotly. So these are the topics that we plan to cover from the technical side. But to really draw the connection between the technical and the conceptual, we will utilize a number of projects. And these are curated projects that our team has gone through, has annotated for the specific methods that the authors of peer-reviewed articles have used, and extracted some examples from these data sets so that we can try and practice on them, both learning about the usefulness of a method, but also being able to understand why this method is being used and how to interpret the results or how to use the results from such a data science approach. Now, these different projects include many different categories, so many different data types, including genomics, epigenomics, metagenomics, transcriptomics, many different types of projects like projects in neuroscience and cancer, infectious diseases, and ultimately this also will be applicable to a project that you can independently develop, because once you understand the data and the needs to transform and understand the data for analysis, you can then actually apply the same techniques to a data set of your own. Now that data set could be found on the public repository, or it could be something that you actually are already working on, so your own data set as well. Okay, so let me pause here and try to address some of the questions. So if you have any questions, please feel free to post your questions in the chat, and I will go one by one and address some of the questions. So the first question that I see here is about different programming that I have used or that we will use in this program. So first of all, I will go through several examples in today's session of processing big data using the Bioinfo platform, how to download from that portal some R scripts that you can modify and use. Then I will introduce you to a number of scripts in R and in Python so that you can see how you can learn those as well. And then the other question that I see here is, can we publish our results in journals using secondary data? So obviously, yes, you can. There are many examples of reanalysis. It's called meta-analysis or reanalysis of existing publicly available data sets. Typically about 95% of the actual data is not being published. It's only the 5% of the most significant, exciting, or relevant findings that people tend to put in the journal article. But that means that 95% of that data is still available for reanalysis. And oftentimes you can actually take and combine multiple data sets, integrate them into a new data set, and ask a completely different question, or even perform the similar analysis using different methods. And that oftentimes will lead to novel insights, might mean that you can find something that could have some significance. And if you do, then you can actually share that in a journal article. Of course, we do not commit to helping you go through the submission. We just can help you by training and offering you guidance in how to develop a bioinformatics project. Okay, so Hadia is also asking if the course will be recorded. Yes, I will come to that in a second. This session is being recorded. It's also being live streamed on YouTube, so you can always find it on our YouTube channel. But I will talk a little bit about the sessions that we will cover in the full program. And so those, yes, will be recorded and shared with all the participants. Are there basic courses for a biologist who doesn't have computational skills, such as R and Python? From Linga Raju. So yes, I think that we live in an era of data science. And so it's critical for anyone who has an interest in applying data analysis to large data sets to learn gradually some of the required data science skills, including coding. And so that's why this program, I think, will be also useful for those of you that do not have the skills to code already, but want to acquire those skills. So that's really what the program is about. I think it's also going to be useful for those of you that do know some coding, but would like to learn a little bit more about machine learning, deep learning, and how to visualize and explore your data. And so I think to explain how the program can achieve all of that in a short time, I would like to demonstrate to you how the program will run and what are the different components of knowing how to ask a question, being able to access big data sets and integrate them, and then how to apply some of these data science skills to also explore and use machine learning on those data, and then demonstrate your results, share your results with others. Okay, so if there are no other questions, thank you, Durba, also for your interest. I think there are many applications in agricultural sciences as well. We do have another session that's going to be coming up about agriculture and the application in agriculture. But today, let's continue and talk about this general field of data science and see how we will achieve all of this during this upcoming program. So the upcoming program will combine 3 different things that you will be able to do. And the first one is interactive sessions. Just like today, you will be able to join a session on Zoom, and one of our mentors, myself, Clinton, or Harpreet, will be guiding these different sessions that you can interact with us and with others, ask questions, get help with some technical challenges that are being, that you're getting stuck with. and then also to get the assignments and the review for your completed assignment. So that will be covered in the interactive sessions. Included will be some self-guided study materials. So these self-study materials that we offer is something that I will show in just a few minutes. And then we will also use some tools. And these tools include the T-Bioinfo platform. Many of you are already familiar with that. It's a logical and user-friendly way to analyze all kinds of omics data, including genomics, transcriptomics, epigenomics, and metagenomics. And then we will turn to our coding portal, where you will be able to try different scripts, including scripts that you've used on the platform. I'll demonstrate that in a few minutes, so that you can understand not only how this platform works, but also what is the specific code and how to modify that code and practice so that you can test whether you understood or not. So I will demonstrate that in a second. So at this point, maybe I can ask all of you, how many of you already have an account on learn.omicslogic.com? All right, excellent, Durba. If you do not have an account yet, please take a moment to register. So let me show you how you can register on this portal. This portal is something that will be important if you would like to try some of the things that we're going to be doing today. That is going to be very useful. So if you already have an account, you can go ahead and log in to your account. If you do not have an account, you can create one by clicking right here. And so let me log in, and I will show you a couple of examples that we will be going through that anyone can try starting with today. Okay, please let me know once the rest of you have logged in. I will give it a moment. All right, thank you, Zara. Shifa, Abir, and Durba. Okay, great. So if you're not yet registered on this portal, please take a moment to register and log in. And then once you log in, you will be able to see right here on top is the menu. And there are some courses, programs, and also right here is your account. Okay, so when you log in, we ask that you go into this account, into your account, and you look at updating your profile, as well as looking right here that you have a subscription. So I will go over some of these things today, but this is just to help you find all of this information on your profile. Okay, excellent. So I see that some of you are registering right now. Once you have registered, again, you can go to the menu and find your email right here. So you can go to your profile. So once you've registered, please put something in the chat box so I know. I'll just give it another couple of minutes for everyone else to join in. All right, thank you, Hidayah and Ayomide. All right, great. Arpita, thank you. Again, the objective today is to talk about the program that is coming up, but also to give you a sense of how things will work. So thank you. Okay. Okay, so great. So now we have quite a number of you already registered. So let's take a look at an example of what I'm talking about when I say we can practice both by running a big data analysis and then taking that, modifying the script, and actually learning how the script works. So if you go to the courses tab, you will see here, first of all, a brief overview of your profile. And then you will see here, you can search for different courses. So let's search for Introduction to Data Science. And you can select either Introduction to Data Science in Python, or you can actually look for Introduction to Data Science in R. And so for today's purposes, I will take an example from R, but you can perform all the same analysis in Python as well, and we will cover that also. Okay, so if you are already logged in, okay, thank you. I see Shifa, Arpita, Hidayah. Okay, so I see a lot of you are now logged in. So again, if you are here, I would like to show you how this will practically work. Okay, and so please go to this first R coding introduction to data science course. And once you are here, we're going to look, we'll take it kind of to the advanced level so you get a sense of what we will be doing ultimately. But if you don't know the basics, there are some basic methods here as well. So let's go right here into dimensionality reduction. So how many of you are familiar with principal component analysis already? Let me know in the chat. How many of you are familiar with what is principal component analysis? Okay, Aditayo, excellent, thank you. Anyone else? Durba, okay, excellent. Abir is not, okay. So for those of you that are familiar with principal component analysis, you know that sometimes principal component analysis can give you a general sense of differences between different data types, or sorry, object types, right? So sample types. For those of you that are not familiar, I will briefly explain. So let's say we have a gene expression table, and this gene expression table contains some samples. We have a header in the table. It has some sample names. These samples belong to different categories, so different groups like normal, like basal, luminal, etc. So this is, by the way, a table of gene expression for breast cancer cell lines. Now, these different subtypes of cancer are important to understand because ultimately they can be treated by different types of drugs more effectively. So if we have a tumor and we want to understand what's the best way to treat that tumor, we need to classify them, organizing them into different subtypes. So here we have 15 different genes and a number of different cell lines, many different cell lines. And our objective here is to understand whether we can see the differences between these different subtypes. Here you can learn a little bit about what this means, and you even have a short video to introduce the full code if you would like to learn afterwards. The second thing that you need to do is you need to load a library. And the library here is important because, as I mentioned, principal component analysis is a type of a machine learning method. It's dimensionality reduction, but it could be used with many additional scripts, not only to perform the analysis, but also to give us a meaningful visualization that will become intuitive as we take a look at the data. So first, we need to load a library like that, and a library like that exists already. And there are many different packages that can add on some meaningful visualization tools for downstream analysis. Now, the second thing that we need to do is we need to use a data set that we can load. And you can see that it's actually really simple. Once you kind of learn to read the code, it's very intuitive. We create an object called input file, and we place in this object some path to the actual data set. Once we do that, we actually translate the input data into an object that will be imported using a specific method. And so here we can use a read table method, or we can use read CSV or read TXT, so different methods to actually load in the data from that file. And then we want to create a matrix. So those of you that are already familiar with R and Python, what is the difference between a matrix and a data frame? Could you please tell me? Okay, can anyone tell me what is the difference between a matrix and a data frame? Aditayo, thank you. A matrix has only one data type, numeric, and data frame has different data types. Excellent. So as we saw, this file contains multiple data types. Here it includes numbers, and here it includes strings. So it includes names, they include numbers and letters. And so that's why the analysis has to be separately done on just the numeric data, but the annotation has to be done using all of the other information. And so here what we're doing in this code is we need to separate one from the other, and then we can actually analyze this data. And so as you go through this example, you can see that the analysis itself is something very simple. It's just one line of code. But this analysis produces multiple types of output based on what we loaded in and how we organize the different objects. And then you can play around with what your output is from this analysis, which is, by the way, a function that you will use that somebody else wrote. And then you can see that you can add on all kinds of useful elements to the output to understand the separation between different types of objects or different types of cell lines, including what genes are most informative for the separation into different groups. Now, to perform this analysis yourself, you can also try running this analysis right here in this coding area, okay? So if you now go to this step number 3, you will see that the input file is given from GitHub. So we have some GitHub link. Then we prepare the full table, separate the matrix, filtering for some data that we don't need. Then we perform the actual object, we perform the analysis, and we plot the result. And once we do that, we can use different types of libraries to actually make this visualization a little bit more informative. And so the cool thing about this is I can run this code right here in the browser to change different things about the code and to see the result right here in the output. So I've run the analysis, and I can try the base R as well as a ggplot output that provides me with some additional useful information. What could be nice here before I kind of continue on is to add some additional information using an additional method of annotation, okay, so that we can actually run. And so that's where we go from the learning about code to an actual challenge. So you've learned the code, right? The code runs, we know that it works. Now let's try to modify this code so that we can add some additional elements that could be useful for visualization. So if I just click on run, I get an error. And you can see that it has an unparsable argument, library gg45, okay? So right from the start, this is not working. So now I have to learn how to troubleshoot this and also include some additional code that now I can make into a working script so that I can make some better visualization of data and understand all of the different outputs about this method. So ultimately, what you will be creating as you figure out this challenge is a visualization of a principal component analysis of a next generation sequencing transcriptomic data set about breast cancer that clearly separates into meaningful groups depending on the angle of the final output, right? And so to learn about all of these different methods, that is why you need to go through the training sessions to know what are the specifics of the code, what are the specifics of the actual method or how it works and what does it mean, as well as some biologically meaningful understanding of what is the context of this data and what is the need for making such analysis, or in other words, how can we actually interpret what we have here in our data, okay? Even after this, you can go beyond the basics and start including some additional information about statistical significance of separation, including clustering, how you can cluster this information, and all the way into feature significance, as we already discussed, so that we can make this even more interpretable. And so that is a huge component of this program, that we will not only look at some of the kind of conceptual things, but you will get to practice and learn how to perform all of these meaningful analyses using a number of different tools for loading data and checking what is in the data, data visualization, including some advanced visualization techniques, principal component analysis, differential gene expression, clustering, classification, and even things like t-SNE. So you can actually look here at also some statistical analysis tests. So you can take a look at some of these lessons. Now, I mentioned in your profile right here, if you go back, you need to have a subscription. So if you want to learn about all of these methods, you don't even have to join the full program. You can use some of these methods in an asynchronous way, practicing independently, and all you need is a starter subscription. So this subscription is designed to be very affordable for those of you that would like to get started and are not ready to join the program. So all you need to do is you need to go to the pricing tab and select the basic starter package. It's only $10 a month, and you can get started with this program already. Now, another important component of this whole program is how did we generate this table, right? So I already talked about generating the table from unstructured data, because most of the data sets that you will find online are actually unstructured. And so to briefly explain the connection between the raw data and this small table that we already worked with in this online example, let me introduce you to our big data analysis platform. Now, I know that some of you are already familiar with this platform, but I will also show you today how you can run an analysis on the Bioinfo platform and download directly the R script that is used for the analysis itself, so that you can open it up right there in the OmicsLogic portal and run the same code that you download from the platform, okay? So please go with me to this other link called Server Bioinfo. If you have created an account, you should be able to use the same login information as you created on the OmicsLogic portal. For those of you that created the account today, you will be able to use that 24 hours from now, so tomorrow. Okay, so if you don't have your login just yet, you can come back and practice this tomorrow. Okay, so here I want to show you a couple of short examples, and these are going to be demos. So we're not going to be loading the data, but I will quickly show you how to go from raw data to structured data and then perform the same principal component analysis that we did here, but directly from the download from this portal. If I go to the first demo right here under transcriptomic data, remember we took here transcriptomic data, I can learn about transcriptomic data analysis conceptually just by following this example. So this example separates the full analysis into 3 sections: pre-processing, mapping, and quantification. Once I do that, I can follow those steps to perform what's called to make a pipeline, build a full pipeline. So as you remember, the first step was pre-processing and it was blue. So I can select Trim-O-Matic, which trims the reads from technical adapters. PCR clean, which then eliminates the amplification using bridge PCR amplification, which is a part of the sequencing process. I can then map the reads that I've cleaned up onto the reference genome, create a new GTF file that contains all the information about the transcriptome, or what are the actual transcripts being expressed in my data set, and not just what's in the database. And then I need to map again the reads onto these annotated transcripts and quantify using RNA-seq using expectation maximization, which in itself is a machine learning process that looks at the statistically significant values for those read abundances across the whole transcriptome. So now that I've performed this analysis, I can also take a look at the outputs. So here I have those genes that we looked at according to the different samples and groups of samples that we already generated and looked at in the previous example. Now, what can I do with this data? We looked at the coding example, Now let's take a look at an example right here, which helps us understand the conceptual link between inputs and outputs that we are expecting to find. So right here, I have an unsupervised analysis pipeline that shows different examples for what we just did, right? So here is a good place to kind of see what are the categories of the different methods that we use typically for exploration of biomedical data, including omics data. And so here you can see that we have a simple pipeline using an R script for PCA. And all I need to do is just download the result, because one of the results is the actual package for R, right? So remember, one of the objectives here is not just to learn about the scripting, but also for effective organization of your code. And so effective organization of your code can be seen right here. Essentially, any executable script typically is stored in a package, and it's written in the language that is used to run that script. And so that is right here. The actual script is right here. I can open the script and I can read what it does. And it's organized very differently from the script that we just edited on the platform, right? How different is it? Number one, it includes some arguments. And these arguments are being passed from outside the script. If you just run this script in R, it's not going to generate anything. There are some arguments that, first of all, need to be addressed to run the script from the command line. And so here you can see arguments for input file, for whether to transpose the file, to center and scale it, and also what number of axes to draw. And the second file that you have is an actual call file that you can run from your terminal to launch the R script together with the specific data set that is explained right here. So this data set has a name and it's a .txt, and then you can see all of your other arguments passed right here in the script line. So this is an important part of what we will learn in this program as well. So we will talk about how the data is generated. You will be familiar with this interface so that you can try out some of these things on your own. You will see that this includes processing of many different types of data. It includes analysis of next generation sequencing, mass spec data, some methods for structural data analysis. And then importantly, you will find a whole section dedicated to data mining, data visualization, and integration as well. And so here you will find an interface that allows you to take huge data sets up to 5 terabytes in size and analyze them using this visually intuitive interface that includes helpful guides on how to get the types of outputs that you need. And importantly, this spans across many different types of analysis, from sequence alignment to variant calling to evolutionary studies to ChIP-seq and whole genome bisulfite sequencing data. So all kinds of methods are included here. And as I mentioned, many of these could then be opened up right there in the script runner so that you can analyze this data and practice the analysis yourself, seeing the correct output and some tutorials with step-by-step explanations on how to get to the desired output. Now, ultimately, we will also go about how to transition from this online experience to actually setting up your development environment right there on your computer so that you can install the right libraries and packages to run the analysis in R or in Python, in Visual Studio or in RStudio. Does anyone have any questions about what we did in this section? Zara, I hope that I answered your question on how this table could be generated. And if you have any other questions, please feel free to write this in the chat so that I can address your questions one by one. Okay, If there are no other questions, then feel free to add more questions if I have not addressed them yet. I want to briefly describe what will be the schedule for the full program. So the program will start on Monday, July 12th, and we will start this program in 2 parts. The first part, we'll talk about the introduction to big data. So what are the different types of data? What are some of the big data challenges and opportunities? How to clean, load, and process data? How to perform exploratory data analysis? And then we'll do some practical training so that we can all try out these methods together. Okay, so briefly, we do have a question from Christina. She's saying, I've just introduced the resources available with the starter subscription. The starter subscription will give you access to the demo pipelines that I just showed and to running the code. The program will actually open up the platform for you to load data sets, and we'll go through NCBI and TCGA and places like that to take curated data sets and process them and then analyze them together. So the program subscription will give you a higher level of access, and it's going to be a little bit more expensive, but I'll talk about that at the end of today's session. Let me know, please, if that answers your question. So the program itself, yes, the starter package is a fully asynchronous training package that you can use to understand how the two are connected and to practice some coding and learn about different types of data sets. The program will include synchronous sessions like today that are interactive, where you can ask questions and get practical help, where you will also get much more in-depth information than is what is provided in the courses. And what we found is that this is really needed to master a subject because so many online courses lack the guidance and the structure. And so that's why we created these programs, to provide with that structure and mentorship so that you can interact with a live person and get unstuck before you get into the program. And the program will be organized into 2 parts. The first part is going to be an introduction, and the second part is going to be focused on machine learning itself. So we'll talk about what is the difference between machine learning and AI, what are the different methods for supervised and unsupervised learning, and then we'll talk about how to deal with the multiple types of models that are available there. I mean, there are so many methods, they all work in a very similar way. How do you effectively choose between them and select the one that will work for the type of project that you have and the type of objectives that you are pursuing in your own project? And that's going to be another important component, is that according to the levels of learning, one of the highest levels of learning is called a project-based learning. And so project-based learning, if you really master something, that means that you can do a project using those methods. So you will learn about a lot of different methods, but to test yourself and to know that you really mastered, you need to try and apply that to an independent project. And so that will be another component of this program, is we will help you apply this to a project of your own and guide you to refine your thinking about when and how, and specifically what does the specific analysis type that you've chosen address in the data set that you've already that you also select. So it's going to be a flexible thing that you can try different data sets and try different methods and then come up with a research proposal and actually try and execute it during the program. So I'll briefly review some of these methods that I've discussed. The first part, working with big biomedical data, will include addressing some of the different types of omics data, including different technologies that are used to generate that data. We will then talk about what is the difference between small data and big data, and what are some additional data challenges that we are supposed to address, and how does that change our analysis practice? How do we go from kind of typical statistical analysis to what is the need for machine learning? We'll then go through cleaning, loading, and processing data. So we'll talk about how to prepare a table, but also how to explore that table to know what to do with it. And we will also address some of the important questions that arise out of exploratory data analysis. So we look at statistical properties, we'll spot batch effects and technical issues that have to be removed and addressed before we actually start the analysis. And then we'll take a look at several examples of how this ties into a workflow. So how to essentially design your workflow from acquiring data from different repositories to preparing a structured data for analysis. One of the questions, and maybe I can ask this of you as well, is, well, transcriptomic data in a sense is easy, right? Transcriptomic data is continuous, and so the numbers are right there in the table. But what to do with data that is naturally categorical? For example, here you have sequences of T, C, G, and A. So essentially numbers 1, 2, 3, and 4. How do we deal with data that is like that to convert it to data that is continuous, that is most useful for some of these machine learning methods? And so the answer to that, of course, is that there are many different ways that we can convert data from categorical to continuous. And once we've done that, we need to incorporate that step of how we convert it to data so that our analysis matches the type of preparation that we did. Now, in the second part, we will talk about data-driven analysis. And that's really where machine learning comes into play, is because we generate a hypothesis from data, and then we build an algorithm, oftentimes not really diving too deep into the biology before we can train a model that could have some meaningful value, right? And so this is a huge change from the traditional approach to data analysis coming from biological experimental design, because we're essentially not trying to test a hypothesis, we are trying to design an algorithm that could mean something useful either for understanding or for a practical use. And I think that's one of the major kind of advantages of machine learning in that sense, because we essentially can not understand everything, but still find use in the data that is generated. So we'll talk about what is machine learning and how is it different from artificial intelligence. We'll turn to unsupervised learning, where we'll talk about different types of clustering techniques and what do they mean, what do they provide us with. We will talk about supervised analysis for classification and feature selection so that we can understand different algorithms, different types of models, and what are they essentially able to provide us with. We will then talk about evaluating the different models and the outputs through model accuracy and validation. We'll talk about ROC curves and area under curve. We'll talk about also how to interpret some of the results from our analysis and how to then use these metrics to select the right model for what we want to achieve. We will then talk about machine learning to get results, right? So one thing is to understand how these methods work. Another one is to actually make them work, right? And that's where you have to build essentially a pipeline and make sure that you are including all of these important steps into that pipeline, and also so that you have a full description of your full code that you can share with others that would like to try and replicate what you have done. So we will address this important topic in biomedical research, which has to do with reproducibility. So what are you going to learn by joining specifically this program? And here we've listed out some of the things that we think are critical for anyone that would like to learn biomedical data science for biomedical discovery. And that includes learning by looking at examples of data visualization and learning about different types of approaches and packages for machine learning in R and Python. Looking at some curated data sets, and by curated, I mean these were selected from peer-reviewed papers. They contain interesting kind of questions and interesting results as well, so we can validate the analysis. You will learn about stages of analysis: exploratory, comparative, supervised, and unsupervised. We'll talk about different types of machine learning algorithms so that you understand the taxonomy and how to read and understand different methods. You'll also learn about FAIR principles of how to describe your analysis and share your workflow. And you will also learn about project design and implementation for independent research. And I will point you to a course actually that we have there that can help you streamline your bioinformatics analysis. Okay, a question from Hidayah. Is it suitable for a beginner? yes, you do not need to know how to use R. If you followed our previous steps, you probably saw that there's some asynchronous coursework on the very basics of R, how to learn about it. So if you are not intimidated, if you're brave and you are up for the challenge, then this is definitely for you. If you feel like this is technically very complex, I would recommend that you first try. So if you have some time today or tomorrow or over the weekend, you can try out some of these asynchronous courses. Some of them are free. And if it seems like something that you can manage, then definitely this is for you. And please let me know if I answered your question. Now, there are some additional benefits that I won't go into right now. You can see them on the screen, but I do want to mention this part of the independent project, right? So as I mentioned, to really learn and master a skill, you typically have to go beyond just reading a textbook and trying out a few pieces of code. So really, it's all about applying these methods that you've learned to an independent project. And so to do that, we've designed a system to help you apply the data that we have curated or that you can find with methods that you will learn about or additional methods that you have heard about or read about to a project where you can modify the settings and the question that you have and essentially come out with a fully independent project. And we have many examples of people that have completed a similar program and have come out with a fully completed project that they have shared and went on to publishing. So let me show you what that means. On the same portal, if you look under courses, you will be able to find a project, or sorry, a course about developing an independent project called Designing a Bioinformatics Research Project. And this is essentially a cornerstone of our project-driven method that helps translate these practical skills into an outcome that is relevant for research. So a lot of the participants that we have here are researchers or are interested in research. And to help them start applying bioinformatics in a meaningful way, we've designed this guide that essentially goes from finding a real interesting and competitive research topic through literature review, to then finding a data set on different public domain repositories, and then all the way to finalizing your analysis and presenting this so that you are ready for publication. So this is all a part of our strategy. And if you go to your profile, you will see how all of these things actually connect. So the first one is training. Right here under your profile, you will find a tab called courses. And you'll be able to see that you have a number of courses that you can go through, complete them, and get a certificate for completing that individual course. So if you click here, it will open up and show your certificate. The second one is, in many cases, it's not enough to go through the material independently. You really have to have someone that guides you and a structure to get you from point A to point B. And that is where these programs come in. Now, you might be advanced and interested in machine learning, or you might be just a beginner and just getting started. And based on where you are, we've designed specific programs to help you get started from where you are and get you to the next level. So if you're ready to join this program, go to LBRN OmicsLogic Data Science. If you would like to just get started and learn about what is going on with bioinformatics, we recommend another program called OmicsLogic Bioinformatics and Computational Biology, a cheap and affordable program for anyone interested to get started. But ultimately, we would like you to be able to design an independent project. And so we have these multiple steps that you have to follow to be able to get there. You have to complete your profile, your bio, earn 10,000 points, which you acquire by going through the different projects and the different challenges along the way. And then you will be able to complete this course that I just showed you, where all of the steps are given and you can submit a project proposal that will be reviewed by our team, and then go through all the steps needed to complete your analysis and present as a research fellow. And so that's why an additional program that we have for those of you that already know everything that there is to know, and you would like to work on an independent project exclusively, we have another specific program called a research fellowship. And this program is designed to actually help you learn and work on your independent, project along the way. So I'm just going to post this link right here. For those of you that are interested in more of a research approach to this whole thing, this is a program that could be between 3 months and 6 months. And during that time, we are confident that you can get to a meaningful outcome, including a full project proposal and presentation. Okay, so that is what I wanted to cover. We have a number of examples of people that have completed this program and have started using some of the bioinformatics tools and data science skills in their research. We do have a lot of these examples already on the website, so you can look at those as well. And if you do have any questions, I would love to answer any questions or concerns that you might have so that all of you can join on Monday and learn with us about data science. So if you do have any questions, please put them in the chat. I will keep this slide on the screen for just a few more minutes. And if you don't have any questions, then I welcome you to join. How do you actually join the program? So again, this program is LBRN Data Science. That is going to come up. Let me find it real quick. Okay, for anyone that has concerns about what will happen once you complete the registration. So what will happen is you will see the program like I do, and you will be able to see inside all of the Zoom links, as well as recorded videos from past sessions. You will see the included courses and projects in this program. And you will also be able to get a certificate for the program after you complete the necessary steps, which includes some of the sessions that you need to complete and some of the coursework that we provide. All right. Great. Thank you, Aviva, and great to see you. Looking forward to seeing Aviva in that cohort. Okay, Christina is asking, would this be the program for intermediate level experience with bioinformatics and its applications to genomics and transcriptomics? Yes. So again, you can join the program as a complete beginner. That's fine as well. But mostly this is for people that do have some sense of what is this data and what they want to do with this data. So if you don't have a lot of experience with genomics and transcriptomics data, this is a great program for you as well. But it will be talking a lot about the technical details as well. So you have to be prepared to, I don't know, Christina, if you know any R and Python already, or are you just getting started? So maybe you could clarify that for me. Hidayah is asking how many hours per day. So each session here, and here you have a schedule, each session is an hour and a half. So an hour of going through the lecture materials and trying out different things, and then a half an hour that we allocate towards Q&A. So it's an hour and a half. Beyond this hour and a half a day, you also have access to our Slack channel, and our mentor community will be able to assist you with any questions. And if you purchase the intermediate level, you can also book a meeting with one of our mentors to help address one of the questions that you have. Okay, so Christina, if you already have some R experience and you're getting started with Python, that this is perfect for you. You will be able to take those skills further as you also learn about the different omics data types and what to do with them. Okay. All right, great. So thank you everyone again for joining. If you do have any final questions, we welcome you to email marketing@omicslogic.com. You can also reach out directly to Mohit at pine.bio. He's going to be overseeing some of the logistics And then we also have Sonalika Ray and some of our campus ambassadors that can help answer your questions directly. So if you do have an email coming from marketing@omicslogic.com, you will be able to respond to that email and actually get a reply. I do welcome you to register. Once you register on the portal and have your account, it will be much easier for you to join the program. And our next session is going to be on Monday in the afternoon, starting at 3 p.m. So looking forward to seeing you there, and have a great weekend, everyone.